Okay, so thanks Ben for the introduction. And hello everyone, this is Sunita. Welcome to the Functional Genomics webinar session. So functional genomics is actually a branch of genomics that determines biological functions of genes and their products. And it is a study of how the genome, transcripts, proteins, and metabolites work together to produce a particular phenotype. But this cannot be achieved without relying on high throughput sequencing technology. So RNA-Seq is actually a very successful application of next generation sequencing technology for genome-wide expression analysis. And this can be used for transcriptome assembly, gene discovery annotation, and detection of differential transcript abundance between tissues, development stages, genetic backgrounds, and different environmental conditions. So the most common application is actually for RNA-Seq is for the identification of genes that show differential regulation under certain conditions. So for this webinar, we will be mainly focused on RNA-Seq technology and especially RNA-Seq data analysis. So outline is this that this session, I will give the brief introduction to RNA-Seq and also will provide overview of RNA-Seq workflow. And Vivek will provide KBase narrative demo. He will walk you through the RNA-Seq workflow in the KBase narrative interface. So RNA-Seq has become the standard method for measuring and comparing the levels of gene expression in a wide variety of species and conditions. And as you see in this figure, over the past decade, there is an exponential increase in RNA-Seq publications. It has actually revolutionized molecular biology. And it is happening because of cost of sequencing is much cheaper now, and there are many new software tools available to address the data bottlenecks. So actually now RNA-Seq methods are available for studying many different aspects of RNA biology, including single cell gene expression, translation, RNA structure, and even this spatial transcriptomics. So it is evolving quite fast. I found that RNA-Seq blog is actually quite useful for the latest innovations in this field. And although RNA-Seq is widely employed, but this rapidly advancing transcriptomic technique can be quite overwhelming. So if someone who is new in this field, I will strongly recommend to check this review article on a survey of best practices for RNA-Seq data analysis. It is by Konisa et al., published in Genome Biology in 2016. So this is an excellent review on all of the major steps in RNA-Seq data analysis, from experimental design to functional analysis. And it also highlights the challenges associated with each step. So typical RNA-Seq experiment, it begins with isolation of mRNA, then its conversion into cDNA, followed by fragmentation and addition of adapters to each cDNA fragment, and like your DNA fragments ends, and then you have to go to sequencing. So sequencing the library of the fragments in a high throughput fashion actually returns as many as billions of sequence reads. And these can be single end or pair end, depending upon the technology and the experiment you have used. And these experiments generate very large complex data set that demand very fast, accurate, and flexible software to reduce the raw read data to comprehensible results. So we are actually in this webinar going to talk about the RNA-Seq data analysis. So once sequence reads have been obtained, these reads must be analyzed with accurate, efficient software. Those are carefully designed to handle this type of the data. And this data analysis pipeline, so it involves, you can divide it into three main tasks. One is alignment. So once you get the raw reads data, this reads needs to be aligned to the reference genome, and then assembly of the alignments into full length transcripts and quantification of the expression levels of each gene and transcript. And the next step is the identification of the differential gene expression among the different experimental conditions. So these are the mainly three particular aligners, assembler, and differential gene expression identification. So let us talk about KBase now. So KBase is open source and it is available for anyone to use. It is free. And in KBase, our goal is to develop an intuitive and robust workflow so that researchers without any programming knowledge can quickly run the tools in the narrative interface. In addition, in KBase, you can also integrate these tools seamlessly with other workflows to generate novel hypotheses. So as shown in this figure, KBase offers a powerful suite of expression analysis tools, starting with short reads in genome. You can see here, you can use the application to align, assemble, and identify differential expression on genes. And then you can also compare the expression data with the flux when studying metabolic models in KBase. And you can identify pathways where expression and flux agree or conflict. So this can be integrated with metabolic models. So in KBase, there are over 300 tools. These are called applications, and they provide quite extensible analytical capability that includes genome assembly, genome annotation, comparative genomics, expression analysis, and metabolic modeling. So application of these first three areas are microbe-centric, whereas application under expression analysis and metabolic modeling, these are domain agnostic. So these can be used on microbes, fungal genomes, and plant genomes. And you can get the details of these applications from this application catalog. So when you click on this link, you can get all the details on these tools. Okay, so in KBase, we have provided Rna-Seq tools based on the standard original and new tuxedo suite to get the normalized full expression matrix and the differential expression matrix of the reads which are obtained from Illumina platform using the reference genome. So please note, in KBase, Rna-Seq analysis is reference guided transcriptome analysis pipeline. So we are not having de novo transcriptome pipeline right now. So it is in KBase, Rna-Seq pipeline is modular. You can see here, it is designed around a common platform-wide data model. So it provides several options to users that they can select, choice of the read aligner and assembler or the differential expression tools. So for this is the original tuxedo pipeline, bowtie 2 and top hat 2 as aligner, cufflinks as assemblers, and cuffdiff as differential gene expression identifier. And for the new one is high set, string tie. And actually we do have ball gown as well, but ball gown is right now demoted in beta mode. And we do have star that is also there, that if you would like to use it, but those are not released in production. These are the released one, which are very well extensively tested. So bowtie actually works for prokaryotes, as you know that it is not does not have splice-aware algorithm, where the high set and top hat they have the splice-aware algorithm, so they work for all the eukaryotes. And rest of the tools, they are generic. Cuffdiff is actually based on, uses different algorithm as compared to DSeq. DSeq based on the counts and Cuffdiff is based on the abundance. So if you would like to use string tie, high set and string tie, and also would like to run Cuffdiff, then you can get the expression matrix, expression data output from string tie and run Cuffdiff on that. And you can also run DSeq and you can compare the results. So this way it is really allows multiple options at any stage of the workflow to choose a tool of your choice based on the strength limitations and your type of the experiment and your hypothesis. So for this particular webinar, we are going to focus on high set, string tie, and DSeq2 workflow. Okay, so the typical Rna-Seq workflow in KBase starts with import of reads and genome, and then reads QC. So once the reads are imported, those reads are actually associated with metadata, and they're created as sample set. And the sample set is used for the QC. And then there is genome alignment, assembly, genome transcriptome assembly, and the differential gene expression. So here you can see apps are implemented in 3 different modular layers: aligners, transcript assemblers, and differential expression identifiers. Because as I mentioned, we use standardized data type in each of the layers, we can support a variety of tools at each level. And it also makes it possible for third-party developers to bring their tools to extend this RNA-seq pipeline. So in green, you see this one, these are all the inputs, and blue are the outputs. So these outputs can also be downloaded, and you can also use, that output, you can also use with other tools and outside KBase. If you are interested to add any tools in this workflow, KBase provides a software development kit that allows the community to add functionality to the system. So Vivek will provide a detailed demo on this workflow that I just showed you, but with the real data set in the narrative interface. But before I switch it to Vivek for demo, I would like to give quick overview of the steps here as modules. So number one module is the import of genome and reads. So you can use, use the public tab in data panel to choose the reference genome from KBase public data. And then you can import the genome from KBase directly from NCBI-RefSeq, JGI fungal genome, or phytogenome. Or you can also import of genomes from your computer through global staging area. You can upload from web to staging, import of GenBank file, and you can also import of GFF3 or FASTA files. Similarly, you can also import of reads from web and from your laptop through global staging area. And from web, there are some apps available. Those are useful to import, single entries from web or paired entries from web and SRA reads from web. Okay, so once you have imported reads, you can group together those reads into an RNA-seq sample set with associated experimental metadata so that you can run the RNA-seq apps in batch mode rather than running on individual reads. And this is really good feature, then you don't need to worry about running each and individual reads. Then next step is the reads quality and processing using sample set. So you will use the sample set for fast QC. And then if for the assessment of the reads, it will provide a report for each individual read object and also for the sample set. And then you can use that sample set to, if it is needed based on that assessment, if you need Trimomatic. And then once you have the Trimomatic reads, then you can again run fast QC to get the assessment. For the alignment here for webinar, we are going to use HiSet2. HiSet2 aligns RNA-seq reads to a genome and discovers transcript splice sites. HiSet runs far faster than TopHat2, and it also requires much less computer memory than other applications. So we are going to use HiSet2 here. And for each sample set, you will get merged alignment set that you are going to use as an input for the next step. And for each individual object, like for each individual read object, you will get individual BAM alignment object. This output can be downloaded and you, and if you would like to browse this alignment, you can also view this alignment by using IGV or any genome browser viewer. You will also get the QualiMap report for each individual alignment set by using BAMQC, QualiMap report. For expression, we are using StingTie, and StingTie in KBase, this can be used in two different modes. One is gene mode, another one is the transcript mode. So generally for prokaryotes, they don't need transcript mode, there's no splicing over there. So we will use gene mode. It is faster protocol. It does not attempt to assemble any novel genes, and therefore the output will only have expression level values for the non-genes given in the reference annotation. For transcript mode, it uses the merge mode, and then it assembles and quantitate full-length transcripts that represent multiple splice variants for each gene locus, and thus it can detect novel isoforms. And again, similar to HiSet2, it will also, like for each individual sample or read object, it will provide expression object, and it will also merge the expression object, and then it will be the merged expression set as an output. It will also provide normalized full expression matrix and 2 units, FPKM and TPM. Again, I'm just giving you an overview. Vivek is going to walk you through all those steps. So next is the differential expression. And for both gene mode and transcript mode, we can use DE-seq2. And you are going to take expression set as an input from string type gene mode for the gene mode of DE-seq2 to get the differential expression on genes. And similarly for the transcript mode, you are going to take the string type transcript mode expression set to get the differential expression on transcripts. And it will provide the differential expression set for the sample set and differential expression for each sample. Now, once you have that, you would like to get some significant upregulated and downregulated feature set and the differential expression matrix based on some statistical threshold cutoff. So that is another app for that. And based on that for cutoff and fold change, you will get the upregulated and downregulated feature set and filtered expression matrix. Once you do have that, you can also get the functional enrichment by running Go enrichment app on the upregulated and downregulated features. And in addition to that, we also provide another app that is called Paper Blast, that is the additional evidence on the candidate differential gene expression from literature. So it uses protein-protein blast to find the similar sequences. Those are linked to the scientific articles. So now I am going to switch it to Vivek. He's going to give you narrative demo. And one more thing before I switch, that help is available. So if you have any question, please feel free to contact us. And there's this link, and we generally are like quite proactive in this, and we will try to help you as much as we can. Thank you.
Great, thanks, Sunita. So I'll pass it over to Vivek. All right, Vivek, I just unmuted you. I'm going to make you a co-host as well. And all right. You should be able to share your screen now.
Let's see, share screen.
Yep, all right, we can see it.
All right, okay. So, hi everyone, this is Vivek Kumar. As Ben mentioned, I work here at Cold Spring Harbor Lab, and I'm a member of the KBase team. So Sunita gave you a fairly good introduction into the kind of capabilities for Rna-Seq and functional genomics in general that you can expect in KBase. So I'll quickly run through for the benefit of many of those who didn't have the opportunity to learn about KBase in the past. I will give you a very quick overview of the different resources with the focus on Rna-Seq, and then I'll sort of dive into the narrative that I'm sharing in front of you right now. Okay. So this is the kbase.us site. You can, as Sunita mentioned, you can sign up and then sign in at the button here. When you go ahead to the about, you will see a brochure which will give you a fairly good idea into the kind of capabilities, the different pipelines, and so the pipeline that we are going to be covering today, the Rna-Seq pipeline. But as you see, the pipelines, they are well connected, and that kind of takes me to the this idea of KBase as an integrated platform, which is integrated by common data types. That's something that I will end up reinforcing at multiple times during the presentation today. So if you see the top pipeline is the assembly annotation pipeline. All of these pipelines, they tend to share a common genome. That's the letter gene in green. And then the Rna-Seq pipeline derives the reads which are processed or can be processed as part of the apps here related to the important trimming and so on and so forth. And then we also have metabolic apps and community apps. There was a question in the FAQ I was just reading across. Some people had a question about community apps. So those are the kind of apps that you can see here in the workflow. And then there are comparative genomics apps, a number of them. The number here indicates the number of apps which cater to that functionality in KBase. So specific to RNA-seq, like you see, the alignment, assembly of transcripts, D-seq, filtering of apps and clustering, and then you can, as Sunita mentioned, you can take this expression matrix all the way to the metabolic modeling, and you can compare flux with expression. All of this is possible because of the common underlying data types that we support in KBase. This is something sort of a distinguishing character of the KBase platform. So getting back to the other aspects of KBase, so webinars, newsletter, something you probably already are well aware of. And for data and tools, so expression analysis is the pipeline that we are going to be covering today. And rest of the data, as you can see, for example, in terms of genomes, again, there were some questions about fungal and bacterial genomes. So if I can quickly take you, so we do support bacterial, archaeal, number of eukaryote genomes. And then I'll also quickly take you through the data panel where we have those genomes. And also we have some sample data for reads and genomes that you can play with if you don't have your own data. And we also have curated narratives, which are also available through the dashboard, which I'm going to show you very briefly. And then in the link here, you will see a number of these narratives. They are well documented and curated. They basically represent the best practices, or we call them narratorials. Basically, they are narratives which walk you through the pipelines as tutorials. There are documentation guidelines. There are extensive documentation sources for how to use KBase in data input, output, upload, download, native interface, and how you can reach us. And in the help, you, like Sunita mentioned, we have very active help desk. Usually we tend to respond within a matter of few days, but you will get immediate response within a matter of few hours, usually in 24 hours. But then someone like me or Sunita, for example, in this case, Rna-Seq will get to those, supporting those queries within a matter of a couple of days. And you can also search the docs here. Okay, so now taking you back to the expression analysis, this link here, data and tools, there's expression analysis. So I already have it up here. So that's the document which sort of gives you a sort of a, so since I did point out a wonderful resource, a publication that covers some of these best practices, but in the context of KBase, we have here all the resources highlighted in one place as to starting with some of the narratorials. You want to run this analysis in KBase, and some of that will become evident as I walk through the tutorial. Things like documentation, active support, and ability to upload, download, some of those things I already mentioned, but I'll also walk you through some of the import and download options which are critical to running a Rna-Seq pipeline. So I'm going to, when I get through the demo, I'm going to quickly go through. So I'm sort of doing a quick overview so that you are not overwhelmed by the variety of apps and variety of ways you can navigate around in the narrative interface. So from that perspective, we have sort of broadly categorized all these capabilities in 3 different groups, which is basically you start, what are the basic prerequisites for you to start Rna-Seq in KBase. So you need to have a genome, or either an existing genome, or you can bring your own genome. And I'll get to those details during the demo. You also need to have your reads. And then once you have reads and genome, you can basically create a sample set and you can run basic quality check. So these are some of the tools that we have. So for quality check, we have FastQC, and then to filter out the contaminating reads, or to filter out the low complexity sequences, or to filter out adapters. We have a variety of tools like cut adapt for adapters, or Trimomatic for fine-tuning of the trimming when you trim out the objectionable reads, which you identify through FastQC. Then once you have imported your genome, your reads, you're done basic quality check. And the next thing you would like to do is basically alignment, so that you can identify your reads in the context of a genome. Again, this RNA-seq analysis in KBase today, and in general, like that's the other question I quickly looked through. We are providing this analysis only in the context of a reference genome. So we are currently not supporting any RNA-seq analysis with the existing transcriptome, or also we are not supporting any analysis if you do not have a reference genome. So you have to have a reference genome all the way through. So for that pipeline, basically, it would be very easily evident that then we need a reference genome to do your alignments. You need basically the same genome to do your build your transcripts once you have the alignments, and then to quantify the transcripts, as well as to quantify the loci in the genes. And once you have quantified, then you can run basically a differential expression analysis. And we have, again, a variety of tools for each of those layers. For alignment, so Sunita actually already went through, so I'm going to avoid repeating that. And then once you have identified the differentially expressed genes, you can take them and project them back to your matrix, expression matrix that you created in the string tie or cufflinks. And that way you can get a filtered expression matrix. And then you can run clustering, you can do enrichment, you can take that expression matrix to a metabolic model and run apps there. So one of the things that I like to point out is the extensive use of data types. And so pretty much most of the analysis in RNA-seq in KBase, you can run on a single sample. But it's really not that useful unless you are basically just interested in running some alignment or just quantifying a single sample. But if you are interested in a sort of a differential expression analysis, which is what we are really trying to do today, you are expected to have 2 or more samples. So that's the reason we, even though you can run the analysis all the way through with a single sample, you would like to have multiple samples in the form of a sample set, which is why you need a create sample set. And then you can run, again, all the apps which you're running until here, which you're running on a single sample. You can run them as a batch on a sample set. So what would be alignment will be alignment set, which will be then fed to a assembler like cufflinks or string tie, and that will generate an expression for a single sample and expression set for a multiple sample set. And also we generate additional byproducts, which you can download, and expression matrices and reports and QC. So one of the things that I like to point out is that we do extensive QC throughout the pipeline. And then we have taken a lot of pain to embed a lot of QC tools. So even when you see the string tie or like FastQC, we actually have embedded additional tools in addition to HiStat itself, tools like QualiMap, for example, for alignment, or like obviously tools like FastQC and other trimming tools. So some of these tools are visible when you look at the output. The outputs are visible only when you look at the output, but because we embedded them inside the core apps. So once you have the differential expression, then you can basically filter and then you can visualize it. So that's the sort of a broad overview. And now that you have a sort of a context for where things stand in the context of specific KBase apps, I'll go on to the next step of walking you through the dashboard, where I'll point you to the narrative that I'm going to be showing you today. So the dashboard is your individual dashboard where you have your own narratives on the top. You have a list of these narratorials that I showed you earlier, which will show up here. You can search for those narratorials or by typing the name of the narrative if you happen to know. Narratives that are shared with you by the people that you collaborate with. We have an extensive concept of orgs, which we'll be discussing in other webinars. but that's where it becomes very relevant for you working as a part of the team to have those shared narratives and quotes. So the public narratives is where I have put the narrative today. So if you type functional genomics, you'll basically see a webinar, functional genomics webinar, and that's the webinar I'm going to be walking through today. Again, for those of you who like to just follow me through on the screen, I think it is perfect, and you can just I'm going to walk through every step of the webinar. But at the same time, if you want to sort of navigate through the narrative yourself, please feel free to go to that place, public narratives, and if you type functional genomics webinar, you'll be able to identify, and also it's first in the list because the latest updated. So it's actually going to be the first narrative in the list that you'll see, public narratives tab under the public narratives section. So I have it already open here. So now I'm going to basically quickly walk through those steps that I identified earlier as a part of the documentation resource. So on the top left, you have a data panel. That's where I will be showing you how to bring your data in. And then there are a number of apps which can be invoked or triggered to run analysis on the data set. And then this is the actual your work environment, your place where you operate on the data on those apps with the help of those apps. Okay, so the narrative that I am going to be discussing today is a taken after a paper. So let me open it here. So there is a paper from Fracasso which basically discusses the impact of drought condition on sorghum. In this case, it's sorghum bicolor. It's a drought-tolerant cultivar. Bicolor comes in a lot of different genotypes. The genotype that we are looking at today is a drought-tolerant genotype. And you might already expected that the treatment that we are subjecting it to is basically whether it's well-watered or whether it's drought-stressed. So for that, you will use this when we come to the treatment. I'll show you. So the nickname we are using is, the condition name is WW for well-watered, and for drought-stressed is DGR. So the reads themselves, they are available at the public website, but they're also, since they're already imported into this narrative, you can just basically directly copy it from the narrative itself. Okay, so Sunita already showed the workflow, and one of the advantages of having a Jupyter interface for our work environment is the ability to document, ability to annotate your results, ability to basically turn your narrative into a sort of a working publication where you can put your input data, your comments, your methods, your experimentations, discussion, results, summary, everything in the context of the narrative itself. And then you can embed the apps which generate that result. So basically what you have is really a close approximation to a publication, except that it happens to be a live publication which you can update at any given time by updating the data as well as updating the apps. Okay. So as I mentioned, the first step is you need a genome and you need reads. So in this case, we have a sorghum bicolor genome. If you go to this little arrow button in the data panel, it will open a data browser. In the public tab, again, I'll very quickly walk through the different tabs. This is my own data that I have generated. This is the data which my collaborators have shared with me, which I can also search. And then there is a public repository of data, curated data that we maintain in KBase, So there are NCBI Rna-Seq genomes. We have fungal genomes from JGI mycocoideum. We have plant genomes again from JGI phytogenome. And then for metabolic modeling folks, we have a number of media data set, and we have some sample data set for RNA-seq here, if you are interested. And also, since our genomes are annotated, when they are imported, they are during the course of import, we annotate those genomes. So we also support public ontologies, and so that's some of the data set that. So if you see. In this case, for example, I am interested in a sorghum bicolor. So I go type sorghum bicolor. And I probably mistyped earlier. So you will see 3 different versions. Again, we maintain the older versions of the genomes. So depending on which genome you are interested in, what latest version of genome, or if you happen to be working on a data set which was analyzed on an older genome, then if you can find it, you can use it from the reference genome that we provide. Now we also, so in this case, I already have added a genome. So we also allow folks to be able to import a genome. And that basically requires you to upload a GFF and a FASTA. And then once you have a GFF, so for example, or a GenBank. So in this case, for example, since I already have GenBank showing up here, so I can upload, I can basically drag and drop the FASTA, the GFF file from my local desktop into this staging area. Basically just drag and drop from your Windows Explorer or whatever platform you have, and or if you have Globus account for those of you familiar with Globus, that actually is also a fast alternative. For some of the data sets, we also provide apps which will also allow you to bring data into staging as well as into your narrative. So when I say staging, this is what I meant by staging. This is the storage space that is allocated to you for a certain period, I believe around 60 to 90 days. And after that period, then this data is basically removed. But once you bring the data into your narrative, into your data panel, it stays there for eternity, basically, until as you like to delete it. And if you want to delete the data or delete the narrative, then it's up to you. But you can maintain the data for as long as you wish. And currently, we do not have any limits on the amount of data you can store. But with the expectation that the data that you are bringing in for staging is the data that you would be using to run narrative in KBase, and that will be data which will be of these types. So the data that we support is again, for the purpose of discussion today, is FastQ reads or SRA reads that I'm going to analyze. As I mentioned, it could be a GenBank genome, or it could be a combination of a GFF genome and FASTA. So when you say, if I select GFF genome, it's also going to, the app that is going to trigger will also give me the option to upload FASTA that goes with that GFF genome. If you happen to have expression matrices already generated outside the system, you can also import them, and then you can run the analysis with those expression matrices, like certain analysis like clustering, or maybe if you want to take that expression matrix to a metabolic modeling, or if you want to do enrichment, any other stuff, you can do that directly on the expression matrix imported from outside the system. So these are basically the relevant data types that for the discussion today: FastQ reads, SRA reads, GBFF genome from NCBI, or like phytogenome usually provides GFF FASTA genome. So for metabolic, for community and others, we have a variety of other options as well. Okay. So for example, in this case, just to quickly show you, since I'm here, if I pick GenBank genome and I try to upload it, it's basically going to launch an app, and it already knows that's the GBF file that I want to bring into from staging, remember I was just now, that was my staging environment. So from staging, now I'm importing it to my data panel and basically to my narrative. So in the staging, it will stay for, I believe, 60 days. And then once I import it, once it becomes part of my data panel, then it can stay there forever. And the data types we support for genomes, again, are, there was a question on the fact. So we support the regular amplified genomes, we support isolates, we support MAGs, bacterial genomes, eukaryotes, pretty much every kind of genome. The source of the genome typically tends to be, JGI phytogem or EBI or NCBI Rna-Seq. As well as you can bring your own genome. Like I said, in that case, the source of genome will be other, and you can basically just keep it other, and it will be able to upload it till the time it's well constructed. So again, note that when we import these files, during the course of the import, the file is parsed. It's also annotated, and so a lot of transformation happens in the file before it becomes a KBase object. A KBase data type object, in this case, is a genome data type object. And that allows that genome object then to be universally used throughout KBase platform. And at that point, you do not need to worry whether the genome that you got was a GBFF genome or your FASTA genome, or whatever the genome it is. Now it's a KBase recognized object which can be used throughout KBase platform. And that's a fairly distinguishing, if not unique, but fairly distinguishing aspect of KBase platform, where we not work with files. That takes away the burden of having to know the types and formatting and all those issues. And you can basically, once you have imported the data into KBase, you can run through all kind of functionalities on those data sets. So these are some of the extra features. It's a common theme throughout KBase that the core features typically tend to be visible, and then advanced features for those of you who are interested in fine-tuning or providing additional context to your data. We provide advanced parameters as well. So since I already have the genome in this case, so I can basically, so the genome that I already imported, it's a eukaryote, it's a JGI phytogem version 13. The size, it shows the number of features, with gene, mRNA, CDSs. It shows the contigs, the 10 chromosomes, and then the additional scaffolds, and the length of the contigs, as well as the corresponding features. So that's the sort of a quick overview of the genome. The source of the genome in this case is the assembly and the annotation that was brought in from JGI. And the taxonomy is another thing. So we associate taxonomy, we associate ontology with all the genomes, wherever they're available. So similarly, we can bring in reads. Again, as I pointed out, if you have reads, you can, for that read, you can bring FASTQ reads and SRE reads. So in this case, if you see, we support a direct download, which is a HTTP or a FTP link to a read in your own server, or a Dropbox or a Google Drive. But for us to be able to access it so that we can import it into the narrative without asking for password or any credentials, you need to make sure that the data is public, at least during the course of import. If after that, if you prefer to make the data private, you can always, once you have brought in the data, you can again password protect it. But for the course of, during the course of the import itself, those links have to support public access to the data. So direct download is the HTTP, and then the FTP link, and then the Dropbox and Google Drive. So in this case, as I mentioned up there on the top, I believe right here. So there's the public FTP site that where we have data, and those are the links for the, so it's a pair-end data. So we have links for the forward and reverse reads for the each of these samples. So the data we are bringing in is basically, as I mentioned, for 3 samples for control, which is well watered, and then 3 samples for treatment, which is drought stressed. So what you see here is the sample number 1, single genome. If you do not know the insert size and standard deviation, that's fine. Some of that you can actually pull out later on from the other apps, like FastQC or QualiMap. You can pull those things out. But at this stage, if you do not know, that's fine. These are optional. And so those are the 3. If you see, there are the 3 samples for 3 biological samples for treatment. Again, for Rna-Seq, there's really no value these days to have technical replicates. So what you're really looking for is biological replicates. And again, if you need to make a for your design, for the course of your experimentation design, if you need to weigh between number of replicates versus coverage depth and so on and so forth, if you have 3 replicates, I think it's reasonably good to do your Rna-Seq analysis, and then you may want to improve your coverage depth. If you have need for like assembly, transcriptome assembly, or like genome assembly and so on, for those kind of things, you probably want to have longer reads, more replicates, and better coverage depth, and because you want to improve the quality of your assembly and annotations. So again, the platform in this case is Illumina. We also support PacBio, Antor, and Nanopore, and so on and so forth. And the result of this will basically give you a quick summary of the read libraries. So these reads that we brought in were FastQ reads. You see let me see if I can scroll through all the way. So it's a FastQ ggypt. But once it's imported into the system, into KBase, it becomes a read library. So those are the 3 read libraries. We have, again, like I mentioned, pretty much for every app that we do, we provide additional context by running additional tools or by providing additional metadata about which provides you additional context for what you're working with. So in this case, the additional context for the reads is the quality of the reads. It's a RAID score 33. Any of the Illumina platforms more recently, starting with 1.8 onwards, they support RAID score 33. For some of the older Illumina below, like before 1.8 or 1.7 and below, they tend to be more of RAID 64. And then we have apps in the system. So if I believe there's an app called, so if I search for quality score, or there's probably an app which you can, let's see if I can. So translate read libraries quality scores. So this you can use to convert your reads from 64 to 33. So that's, again, so these are some of the metadata about each of the reads, the read count, and like quality, maximum qualities, mean quality, length of the reads, and so on and so forth. Okay. And similarly, I bring in the reads for treatment, and that gives me 6 samples. Since I want to operate on this all the 6 samples as a batch, I like to create a sample set out of it, and I'll then annotate those samples with conditions, with treatment conditions. So I have 3 samples which are control, which are well watered, WW, and there are 3 samples which are drought stressed. And here I can provide additional information about the pair end or single end, and I can provide additional context for the samples, the paper, library type, and the source. So this can be useful information for your collaborators who are working to analyze the data, so they would know right away what kind of sample set that that you have in place for your analysis. So the result of this is basically a sample set. Since the reads are already there, so it's basically wrapping those reads into a set. And that set is what we are operating with. So it's basically a summary of those 6 libraries. So now that you have this, now you have a starting point. So now we have genome, we have sample set. So now we can basically, get busy with the pipeline. So the first step is the fast QC to understand the quality of the reads. So in this case, all you need to provide is the sample set, which is the sample set that I generated up here in this app. So that's the object that I provide, and then it gives me a FastQC report for the all the 12. So if you see, there are 6 samples, and every sample has a forward and reverse reads. So it gives me 12 reports. If I open it up, since it's a detail, it's a long report, so I can open it up, I can download it. For a lot of for pretty much every app, wherever possible, we try to provide downloadable resources, the kind of resources that the app or the tool, the underlying tool, generates by default, so that you can also look at the native version of the output, as well as we provide all kind of annotated, visually insightful ways to understand the data in the context of KBase. So in this case, I already have opened up the FastQC report. So these are the 12. So if I look at any of them, let's say the report for first read, for forward version of the read, it tells me how many sequences in the read. The sequence length is approximately 100, the GC level, and primarily what I'm really interested in for practical purposes is the per base sequence quality. So if you see here, again, the scores are based on Fred. So the red line in the middle, that's the median value. This is a interquartile. So this is basically box whisker plot. So it's a interquartile range, 25% to 75 percentile. And then the bottom line is 90 percentile, and the upper line is 10 percentile. So that's your regular statistical plot, box whisker plot, that it generates. Now, you would like to have most of your reads if I traverse or if I span the read from base pair number 1 all the way to 100. So as you know, the sequencing machines, they tend to degrade the quality of the output as you traverse further along the read. So it's common to find some of the later bases in a range which is somewhat objectionable. But again, till the time the majority of the base pairs are in green, this is basically are good to go. A sort of quick context for at what point does it become green, or what point it's warning, or what point it's failure. So if I'm not mistaken, if the lower quartile for any of the base, so if that is less than 10, or the median, so this is the median here, right, in the red line, if that is less than 25, then it will sort of start giving you warning. And same way, if the median is less than 20, which is a little bit more stringent, then it will start giving you error warnings, as opposed to just regular warning. It will give you more like failure warnings. But you can get a quick view of the FastQC quality of the reads by looking at this. Then there are things like what's the most common quality, or the mean sequence quality for your reads. So if you as you would have expected it, the majority of the reads are around, in this case, around 37. So it looks reasonably good. Again, traversing the reads from 1 to 100, this is looking at the ATCG scores for each of the bases. This is averaged out across all the reads. This is again the GC content, GC distribution. It's as opposed to a more traditional normal distribution. There's a hump here in the distribution. And contents, there are no and contents in this case. You can mask if you're masked genome, if you're a soft mask or a hard mask genome, based on that, it can generate some of those end reads. Then there's a distribution of the reads. Majority of the reads are length 100. There's basically in this case no reads of length 99 or 101. Duplication, it shows the percentage of the reads which are duplicated. So as you would have expected, majority of the reads, they have a 1 duplication level. Basically, they are not duplicated. And then some of the reads, there's a certain percentage, around 20%, which tend to be in this particular data set. Again, remember that this is a small data set that we are working with for the purpose of the demo. So usually will like in this case also you'll notice a error flag. Again, the error flag does not mean that you have to then throw away the reads. It's just something to look at what is the nature of the duplication. And the duplications can arise from like the PCR that went into the library prep up. So there are PCR artifacts, so there are biological duplicates, which are basically the obvious collisions that happen when different copies of the same sequences are randomly selected. So, and also sometimes if you want to observe some of the lowly expressed transcripts, then it's common to then over-sequence the library, and in that case, some of the higher expressed transcript, they get over-sequenced, and that will generate some of the duplicates as well. So there could be a number of reasons you can have higher duplication level. This itself does not right away rule out the quality of or the usability of the library. Okay. Then the overrepresented sequences, again, certain sequences, if they tend to be enriched, it will point to you what those are, and you may want to be interested. You may want to look at it like, okay, why I have maybe what led to those. You may be interested in examining those enriched sequences further. When the prep up happens, you usually will have some kind of adapters. If the adapters are left inside the reads by your library preparer, then those adapters will show up, and it will also show you what those adapters are. And then the percentage of the adapter, if it's significant enough, then you may want to trim out those adapters. Mostly it's going to be in the beginning or the end of the read. And similarly, there are k-mer content. If there are certain k-mers which are more enriched, typically I believe it gives you a warning if it's the p-value for the k-mer is I think less than 0.01, and then if it's 10 to the power minus 5, then it gives you a failure. So these are some of the things that that you can learn by running a fast QC about the reads that you brought in for analysis. Okay, so in this case, since we are generally satisfied with the quality of the reads, so we subject the reads to a alignment, and in this case, high set is what we are running through. So the genome we have is sorghum bicolor already imported, and the reads, and usually when you are running multiple Rna-Seq analysis or maybe multiple alignments in the same narrative, you may want to specify the suffix, so that way you can uniquely identify. So in this case, since I already have one Rna-Seq analysis, one high set app, so I basically just say underscore high set. It will basically put a underscore high set suffix in front of all the alignment objects that I will generate. So which in this case is basically 6 alignment objects. Here you can also provide, as I mentioned, the phrase 33. 64. So in this case, it's by default is fit 33, which is what you usually would expect with the more recent Illumina reads or other reads. Most of the other options, unless you have a specific need, if you are not aware of the options, if you don't understand them, then that basically means that you can just leave them alone. Unless you have a very specific need, in that case, you can customize those options, which is the reason we tend to hide those. And the result in this case is alignment apps, the alignment objects, and an alignment set, which contains those 6 objects. So that's why there are 7 entries, 6 of them alignment objects, and 7th one is the set. Also, like I mentioned, we do additional QC. Unlike some of the other platforms out there, we, as I mentioned, we don't have to run those additional QC tools. We have taken the trouble to embed that inside the core apps itself. So you'll have the added advantage of looking at your alignment quality as a part of this app. So here's a summary. I also have opened up. So if I click on the view report in separate window, for the sake of easy exploration, I already have opened it up in another window. This is the output of the BAM QC. There are again a number of QCs that are out there. In this case, also the tools, the BAM QC, Count QC, a number of other QC, BAM QC is what we are looking at here. So a quick look will show you there are 6 samples, 2 groups, the mean sample coverage, the mapping quality of the alignments, and for each of the individual samples, the same thing, and insert median size. So if you remember, I was telling you about the insert size. So this is where you can get an idea of the insert sizes. And this is a broad way just to visually look at the context of where the 6 samples stand with respect to each other. So this is a principal component analysis. It's basically trying to project all of your reads, highly dimensional, into a 2-dimensional screen. So you have your first principal component, which captures the majority of the variation, is really what you want to look at. And then to some degree, you want to look at your second principal component, which then captures the remaining variation. Obviously, there are additional principal components which will capture the leftover variation, but these 2 will capture the majority of it, and PC1 will capture the most of it. So it's not terribly dissatisfying. I mean, we have these 3 libraries which are on this side of the screen, and there are the other 3 on this side. It's until they are all like randomly spread out, then I may want to look at, okay, why I do not see some kind of a common pattern, because I may want to, I may be expecting some sort of underlying theme for my treated samples versus my control samples. Again, depending on the impact the treatment had on those number of genes, you may see them closely clustered if it had a really huge impact, or maybe not. Right? So it gives you some idea of where those samples stand with respect to each other. Okay, and I'll show you a few interesting. So again, this is in interest of time, I'll quickly run through. So, for example, in this case, this tells you what fraction of the genome. So if I say that 42% of the genomes have a coverage of 2.5, reasonably low coverage. But as you look for higher and higher coverage, the percentage of genome with such a high coverage will obviously tend to diminish. So it gives you some idea of what percentage of genome has what kind of coverage. Again, if you can look at the GC content, it goes from, again, as you traverse the read from 0 to 100, how the GC content varies for the 6 samples. Again, where some of the bases have been clipped, usually we would expect on the left side and the right side of the read. Mapping quality, insert size, again, the position in the reference, Again, this is a relative position. So instead of 0 to 100, it's a 0 to 1. And that's the insert size for the reads. Remember, the read length itself is 100, but the insert size for the read fragment. And then there is the histogram of that. So again, this gives you a quick overview of the some of the information actually sort of overlaps with the FastQC, and some of it does not, because you have additional context with the benefit of a reference genome. Okay. So, and then we also have wrapped all the samples together as opposed to having a QC for each of the samples. So that's why we have multi-sample BAM QC. So now we have alignment, we have QC of the alignment. The next step is to start building up the transcripts by looking at those alignments, and then we would like to quantify those reads in the context of the reference genome. So we can quantify the gene expression. So the high set, this is the, if you remember, the underscore high set was the suffix I had used for my sample set to create the alignment. So that's the alignment set that I have. And there are some advanced options. Again, I can provide the suffix for string tie. So in this case, underscore string tie, I can give it any name, basically. That's how I uniquely identify my output object. And then these are default options. One of the options is a make novel transcripts. Sunita mentioned that as well. And we have an app further down which discusses that. So the result of that in this case is a transcriptome assembly for each of the 6 samples. And that's the transcriptome assembly object. We'd identify it as Rna-Seq expression. And the set of that will be Rna-Seq expression set. We also have additional data types, because now that we have the quantification of the reads, we can capture that in the form of a matrix. So we have FPKM matrix, and then we have TPM matrix. So FPKM is the fragments per kilobase of read for per million, per kilobase of read length for per million of reads. And TPM is transcripts per million. So for those of you who already are well aware of it, you may want to use, you may already be aware that TPM is what you want to use. For those who are somewhat new to Rna-Seq field, again, there are FPKM. You'll often find in the literature things like FPKM or TPM. Sometimes you may also find RPKM. So FPKM is what was traditionally used. It was very popular in the context of the original tuxedo pipeline for top hat and cufflinks. But the FPKM, the way it normalizes the, again, so what does really FPKM or TPM do? They are basically ways to normalize your expression so that you can normalize them with respect to the read length as well as with respect to the depth of your library. So FPKM happens to normalize the reads with respect to the library depth first, and then with respect to the read length. TPM does it in a converse manner. It basically normalizes with respect to the length first, and then with respect to the sample size or the read library depth. And that's the reason if you are comparing multiple samples in an experiment, you want to use TPM because it's the final results are normalized with respect to the sample size. So that allows you to compare the samples on the same footing, because all of them have been normalized to the same library size, because that was the last normalization that happened in TPM. So usually, even though you may see some of the literature studies done with FPKM, TPM is really the preferred choice. It allows, especially if you are comparing multiple samples. If it's one sample at a time, FPKM will do the same thing as well. Then it's okay. RPKM, like I briefly mentioned, it stands for reads per kilobase of transcript per million mapped reads. So basically the way it works is if a single end library, one read means one fragment. But if it's a pair end library, then one or two reads may correspond to one fragment depending on if it's a 1 or 2 mapped. And that's where EpiChem actually is slightly better in some terms of capturing the map, or in terms of notation of the reads. Anytime you communicate your normalizations, EpiChem is, because it does not count the same fragment twice if two reads map to the same fragment. It barely counts it only as once, which is what you want. And then TPM, like I mentioned, it allows you to be able to compare your samples all on the same footing across multiple samples. So the libraries that the matrices that we generate are TPM and FPChem. FPChem for the, for backward compatibility, but TPM is mostly what we prefer to use. Okay, so this is a quick overview of the output library. Again, one thing I skipped over earlier was the, let's see if I did. No, actually, I actually did show you. So the QC report. Now for string tie, the default output of the app is captured in the string tie _ result.zip, which you can download onto your local desktop. But for those of you who just want to quickly look at the visually examine, you can just look at each of the transcriptomes, individual transcriptomes. So in this case, for example, if I click on the high set string tie 1, like this happens to be for the first sample for well watered, I already got the visualization here widget in KBase. It allows me to look at the FPKM values, the TPM values. Again, they are +1, so because log2 of 0 does not compute, so they are all with respect to +1. And then there's a histogram where basically we are binning those values and showing the number of genes with those TPM values. And it's a very interactive histogram. You can change the size of your bins, number of bins, and you can draw some cutoffs. So it's a neat widget to explore the output and interactively visualize it. Again, it's some of the value add. At every step in the pipeline, we provide value add, as opposed to you figuring out ways to do those things with tools outside. We try to enrich as much of those additional value add by embedding those tools into the pipeline. One of the other things, so now this is a exploration of the individual sample, but the TPM matrix contains all the samples and all these 6 conditions. So here you can look at the min max for each of the conditions. You can look at min max for each of the features. So, and as I mentioned, we annotate all of our genomes. So that's why you will also see annotations captured here in the function column. So these are the features. These are the min max values across all the treatments for that particular feature, and the average and standard deviation and so on. So again, this is a TPM matrix, so that's why we don't capture fold here. It's just the TPM values. The fold we will capture in the next step, which is now D-seq. So now that you have all the expression quantified for all the 6 samples, now we want to basically look at how differentially they are expressed with respect to each other in those samples. So the expression set was the output from the previous app, string tie. Now this could be a cufflinks, or it could be string tie, right? And same way, this could be, in this case, D-seq or it could be a ball gown, could be cuff diff. So that will be the corresponding step. So here's the expression set. I'm interested in genes, not transcripts. For the purpose of capturing differential expression, I like to run all pair conditions. In this case, there are only 2 conditions. So it's basically going to be, so 2 conditions are A and B, well watered and route stress. So those are the 2 comparisons. If there are 3 conditions A, B, C, then it will generate A versus B, B versus C, and A versus C. It will generate 3 different combinations of the folds. So that it allows you. If you want to only selectively run those comparisons, then you can unselect that, and then you can choose those partial comparisons. Okay, so here's the output of the D-seq. It generates a differential expression matrix that captures all the differential expression contents. There were 34,000 features with that it's computing fault for. The results are captured just like the DSeq tool will generate in the DSeq result.zip, and the plots are captured in the DSeq plot.zip. Let's see if let's see. So there's a volcano plot. If I like to look at the differential expression data, the log2 fault chains versus the Q value. Now, remember that this is a differential expression for thousands of genes. So, and we're trying to see which genes have differentially expressed across thousands of genes. So every gene, there's a possibility of a false positive. So that's the reason we compute Q value, but that will be captured in the later step. So in this case, you can just see those Q values, and you can draw cutoff, and then you can identify what genes are you can basically maneuver these values. We are in the process of refining some of these widgets. One of the widgets we plan to update in the near future. But basically, this is the place where you may identify the differentially expressed genes. Okay, let's see. Is there anything else here? So the next step, now that you have identified you have created your differential expression matrix. Now the next step is to filter out those genes. So I have the expression matrix that was generated from string tie. I have the results from the DSeq. So what I'm doing here is I'm projecting the results of these DSeq differentially expressed genes, and I'm projecting them on all these features. Now, the string tie has all the features for the entire genome. So I'm projecting that, and then I'm drawing a cutoff, in this case, a fault change cutoff of 2. Remember that this is a log fault change cutoff. So on a log 2 scale, it's a fault change of 2. So which basically means in terms of absolute value is 2 raised to power 2, which is 4. So anything more than 4 times or 1/4 is what we're interested in. And this is the Q value, which I referred to earlier, the adjusted P value, 0.05. And I can provide the name of the features, which will be filtered out, and the filtered expression matrix. So this expression matrix is a subset of the original expression matrix, which only contains the features which are showing a fold above a cutoff of 2 on a scale of log scale of 2. And for a cutoff alpha value, Q value cutoff of 0.05. So that means I have a smaller subset of the expression matrix as a result. So that's in the result you can see, since I provided a cutoff of 4, absolute value 4. So I have 27 up filtered and up regulated and 45 down regulated features, and as well as the corresponding expression matrix, which captures those 45 plus 27 features inside this expression matrix. Now I can run similar analysis for transcripts, and that's what I was referring to earlier. And if I am interested in looking at de novo, some of the transcripts which, so far what I was interested in was looking at the transcripts which are recognizable in the context of the reference genome. But if I am interested in looking at novel transcripts, then I can enable that option, and it will also try to identify novel transcripts which are good for gene discovery, transcript discovery, and provide you candidate transcripts and loci to look at. And that extended genome based on the additional novel transcripts is captured here, and that's the SBI_STR_genome. That's the genome on the left side. It generates here. It's a non-taxon because it's generated by the app. It's not a taxon that we are associating from outside. And the transcripts, the newly generated transcripts, they will have a prefix in front of them, so that we can identify what transcripts are newly generated. And then the rest of the analysis is just like D-seq. You can run the same way, just like we did earlier. Okay. So now the next step is, now that we have identified the genes of interest, we want to see how well they are enriched. So there's a Go enrichment. So in this case, I'm using upregulated features. So that's those 27 features. So I'm looking at those 27 features. It's a very small set, so the p-values, q-values won't be as unnecessarily as, again, it depends on the background distribution as well as the p-value, but may not always be very significant. But that's the subset we are playing with. So there's a feature set that I want to look for enrichment against the background of the genome distribution. Parent-child term propagation is how I propagate the. So I'm not only interested in those particular terms, but I'm also interested in the parent terms of the gene ontology when I'm running my analysis. So I'm propagating the gene ontology tree all the way up for the course of running the Go enrichment. So it captures the root terms as well, or the higher or intermediate nodes of the Go tree as well. Since I'm interested in overrepresented or underrepresented, so I have right-tailed statistical test and then the results. The results will show you the enrichment table, the terms which were enriched, whether they are from molecular function, biological process, M, F, or cell compartments, the description of the Go term, number of features in the feature set, and number of features in the background genome for that term, and the corresponding p-value and the corrected q-value. We also generate a number of additional files, a number of other tables, and permitting time, I will run through some of that. So that's the place where a number of research queries will end, where you are able to eventually converge to the genes of interest and what those genes are. You have characterized them, and then these are the genes which were upregulated as a course of my drought stress treatment. We can also run some additional analysis for looking at the some of the homologs for genes of interest. So if I am interested in just to understand maybe what else can I learn about this particular gene of interest, since it seems to be upregulated with a high fold, and I want to learn more about it. So there's a app called Run Paperblast with the protein sequence. So in this particular case, let's say this is the candidate gene. So I can go to my my genome, to my features, and I can type the particular gene, and then I open it up. It will show me the some of the attributes about the gene, as well as the amino acid sequence, which I already have captured here in the interest of time. So if you see the for that particular amino acid, that particular feature that I just showed you, which I expect to be upregulated, or a feature of interest based on my D-seq analysis, I can provide the amino acids for that feature in the Paperblast. And so that's the corresponding amino acid sequence, protein sequence. And then it will generate a hit of the some of the articles or publications which have discussed proteins which are homologous to this particular protein, purely based on the amino acid sequence. So it's running basically a protein blast. and then it's identifying based on the homology, it's finding any of those homologous proteins, and then it's capturing those publications of interest. So in this case, for example, it captured a zinc transporter. So this is something that we would expected for as a part of the upregulation because it's involved in the salt stress. So now this particular one in this case is for Aetheliana because that's where a lot of information exists on this particular protein in Aetheliana. So it gives you an additional insight about the genes of interest by running paper blasts, just like we did for functional enrichment. So, and similarly, you can do additional features of interest. You can run them through. So, so that's a sort of a quick overview. Ben, how much time do we have here? Depending on the time, I can stop at this point, or
yeah, as much time as you want. I mean, we can go for another 30 minutes if you wanted to, but yeah, maybe good to kind of wrap up and start to get into some of if there's any unanswered questions. But yeah, you can keep going if you want.
All right. So let's see. I believe I've captured most of what I could show in the narrative interface. There are a few things as I pointed out, the tools generate their own output. And that output, let's say, for example, in the case of string tie, the result is captured here. I'm waiting for it to open up. So result is captured here is string tie underscore result.zip. And for the transcripts, it also has a deno transcriptome string tie merge. So something like that, if I like to visualize, I can download it and I can visualize it outside. So I'll probably take maybe around 5 to 10 minutes, and then we'll try to see if we can use the remaining time for the QA, as well as the QA will continue after the session is over as well. So, and the reason I want to sort of run through some of that stuff is, so some of you, if you are interested in then exploring the results further outside KBase, because you had this detailed analysis generated by the tools, and we capture that analysis as well. And in addition to providing you these visual insights and detailed reports, so I will see if so I have already downloaded the results in my local desktop, and then I'll quickly run through those and provide you some additional insight into the output of those apps. Okay. So I'm now going to see if I can change my screen. So I'm going to start sharing again, so just bear with me for a minute. Okay. So can everyone, Ben, can you see my screen? Can you guys see the screen?
Yes, we can.
Okay, yeah. Great. All right. So genome, if you remember, the genome was one of the things. So let's say now I have a genome. So I can download that genome. Let's see if I can quickly find it here. So any of these reads, so the genome I have here, but any of the reads, for example, if I click on this ellipsis, horizontal ellipsis, 3 little dots, I can go click download, and then it will allow me to download that as FASTQ. I can send it to staging. Those are the two things that I'm probably interested in. Some of the developers may be interested in looking at JSON just to look at the underlying reference, the object reference in JSON. But basically, we provide this metadata for the objects, as well as we allow abilities to be able to download and send it to staging. One of the things of interest is what kind of apps you can run on the reads. So if I click on this little button arrow pointing this app as input, it immediately opens up all the apps, not just in Rna-Seq, but also outside Rna-Seq as well, like communities and just general utilities, wherever those apps may be applicable, which can work on these reads. Now for sorghum bicolor, which is the genome that I had, I can, if I click on these dots here, and then click on the download button, it will allow me to download a GenBank or a GFF and FASTA. Now, if you remember, when we imported it, we also allow you the same formats. We allow you the GenBank and GFF FASTA. So we allow both. Now this sort of closes the cycle. This is the download aspect of it. So you can download a GFF or a GenBank. So, and that's what I wanted to show you. So I already had downloaded this as a GFF and FASTA, and the GFF file, and there's so I actually dropped out. Maybe I believe the FASTA, I removed it from here for some reason. So here, for example, you can see the FASTA right here, and the corresponding GFF. I added some extra data here, but not captured originally. But those are the two things you'll get. Now, similarly for string tie, the results are captured in string tie result.zip. That's the folder. If you see, there were 6 samples. For each of the 6 samples, for those of you who are familiar with the string tie tool, you can recognize those files. For those of you who are new to it, I'll very quickly run through it. There's the exon quantification, there's the intron, there's the transcripts. So those are the 3 different quantifications. If I open up one of them, I can look at the corresponding transcript ID, the location on the reference genome, the transcript name, the number of exons it maps to, the length of the transcript, and FPCAM. That's the default output it generates. Now, in the system, in KBase, we also convert it into TPM. So that's the value add that we provide, some of the things that we provide in addition to what the tool does. And this is the corresponding JTF that you get from this string tie for this particular sample. So that's how we generate for all the 6 samples. So again, here also there's some data that tool provides, and then we add some additional value. There's the TPM matrix. Maybe I should open it up in Excel, maybe make it easier. So those are the feature IDs, and then each of them is the 6 samples. For each of the 6 samples, this is the TPM value for those 6 samples, normalized. So that's the string tie result. D-seq result, again, there is the fold values, because I did not show you earlier the fold values, so that's why I'm going to show you quickly here. These are the fold values: Q value, the corresponding P value, the log fold change, and the gene IDs. So all of that information, just like you would expect in a tool, we also capture that. Same thing we do for transcripts, and then we also have the raw counts. They are not normalized; they are raw counts. Okay, and then there are plots. There's a PCA plot. I already showed you that in the context of FastQC and QualiMap, and then the p-value plot, and then the dispersion plot. This is basically for those of you who have worked with microarrays. You may be familiar with the MA plot, is what they call it. This is the average of the expression. In this case, it's on the log scale, and this is the log fold change. And what you're looking for is for any 2 samples at a time that you are looking at for the MA plot. You're looking at the trend of the samples. So if more or less, if the distribution is uniformly or symmetrically scattered around it, usually you would expect your distribution to be. And then the ones in the red are the ones which are significantly expressed for that cutoff. 0.05, I believe, in this case. So it gives you some insight into, again, if there's any if there's any distortion in the underlying data for 2 samples at a time, you can see it. So in this case, obviously, more or less, it is somewhat symmetric. So it's good to go. So these are some of the additional results from D-seq. Then filtered enrichment, I believe I showed you earlier in the UI, but those are the filtered expression matrices. Let me open it up in Excel. And these are the, again, and this is a smaller subset. If you see, as opposed to those thousands of features. We only are looking at 73 features. Captures all the up and down regulated features and the corresponding TPM values, expression values. So again, this is a value add tool that we provide in KBase. It's not a part of a standard tool outside. It's something that we provide in KBase. Up, down, up regulated, down regulated, as well as the overall. So these are the down regulated features, up regulated features, and then all the total features. And so I captured genome, string tie, and D-seq. The other thing that is left is the Go enrichment. So these are the files for Go enrichment. These are function enrichment. These are the terms. If I showed you earlier, if you remember the table, these are the 47 terms which were enriched. The number of the features annotated with that term in the feature set, and number of features in the background, and the corresponding p-value and the corresponding q-value. So that's obviously the first thing you want to look at. Then I have some additional files here to show some information about the genome. How many features in the genome have the term. So 37,000 features are annotated. Total number of features is 128,000, which includes, why 128,000? Because 37,000 is the genes. We also have mRNA, CDS, and so on and so forth. So that's why the total number of features. And then these are the feature IDs. And if you see here, that's what I'm pointing to, CDS and others. The ones which false they are not annotated because they are CDSs, only genes or mRNA is annotated. Then this is the mapping between the feature ID and the corresponding Go ID. So so here, if you will, the primary key here is the feature ID. And then for every feature ID, you have one or more Go IDs associated with it. There's also a mapping of the Go tree, where the first term is the parent term and the other terms are these children terms. So, and then let's see what else I have. And then there's a Go ID, and then so in this case, the first term is the Go ID, and then all the other terms are the feature IDs, which are annotated for that Go ID. So now this one was for the entire genome. So that's why it's a bigger file. This one is only for a subset. So they are the Go IDs and the corresponding feature IDs. Okay. So I believe that does it. So I think at this point, I have walked through the ways to import, export data in and out, run the pipeline, do a quality check, as well as how do you get insight by looking at the output results. And also, how do you get insight by looking at the files that we generate as a part of the default tool output, as well as some of the added files as a value add that we provide in the tool output, in the file output as well. So I believe at this point I'll probably stop and I'll take questions.
Thanks, Vivek. I'm just looking at the document to see if there's any open questions. It looks like Sunita answered them all. So great job, Sunita, for answering those in the background. Yeah, I think we can go ahead and wrap this one. Looks like a lot of folks have logged off. So we've still got about 50 people on the line, but so we'll once this recording finishes rendering and all of that, we'll share it with everybody. Please take a minute to fill out the webinar feedback form. I'll just drop it into the chat really quickly. And yeah, follow us on Twitter at DOE KBase if you can. And yeah, we'll keep in touch about the next webinars next week. We've got one on microbiome metabolic modeling, and We've got another one the week after that on phylogenomics. So we're going to be doing a lot more of these digital webinars for you all. So keep in touch with us and keep following us online. Thank you.
So I'll quickly take, so I believe there's still one, I see one question open. I believe most of the questions are answered. There's one question needed to rest. We can answer that later. There's a question, I think, can I use an assembled genome for this analysis if I do not have raw reads of the genome? I'm not sure if the raw reads, it means you need to have an assembly. This is a reference genome. So you need to, now technically you can run apps like HiSet, Top Hat, or Star just with assembly. And you can even run with even the string tied cufflinks also just with assembly without annotations. But in KBase, we actually require you to have a genome, which basically means that you have to have GFF annotation captured in addition to your assembly. So you need to have a reference genome which has both assembly as well as annotation, and if that's what the questionnaire meant. Now, if you do not have those raw reads, technically you can generate transcriptome assembly outside the system. You can still run, again, technically you can run bowtie on the transcriptome. So basically you have your raw reads, you are merging those raw reads, you are creating a maybe Trinity or something, you are using a de novo transcriptome assembler to generate your de novo transcriptome, and then you can basically map that against that transcriptome using bowtie, which is an ungapped mapper. And in that case, again, you don't want to use Top Hat, HiSet, or Star, you want to use bowtie to map your raw reads against a transcriptome, de novo transcriptome. But we do not support that in KBase. But just for the context of this question, I provided all those different scenarios that what you can what theoretically can do.
Thank you, Vivek.
All right. Sure. So I appreciate everyone for taking time, as well as I'd like to extend my thanks to Ben, Alicia, Jack, as well as Dan Olson, who is at this point making sure that the narratives are up and loaded for everyone, and the system is up and running. And thanks, everyone.
Thanks so much, Vivek.
Thank you.
Thanks, Sunita.
Thanks for watching this webinar. For more webinars and tutorials, subscribe to our YouTube channel. You can see announcements for upcoming webinars and recordings for previous webinars on our website at kbase.us/learn. Let us know in the comments what content you'd like to see in future webinars. Be sure to follow us on Twitter at DOE-KBase. And if you have questions or encounter an error when using KBase, please contact the help desk.